Hi, it's quite an honor to present to the Brain Initiative Transformative Non-Invasive Imaging Technologies session, and I'm going to talk about molecular imaging applications of magnetic particle imaging. My name is S.Conolly. I'm a professor at UC Berkeley, both in bioengineering as well as in electrical engineering. And I'm real excited to present our work today on magnetic particle imaging. So as I said, we're going to talk today mostly about applications to molecular imaging using this new emerging technology, magnetic particle imaging. So magnetic particle imaging is a new imaging method that reuses MRI contrast agents, SPIOs, which are super-paramagnetic iron oxide nanoparticles. Some of these are FDA approved, but not all. And so just to give you an overview, we have these 25 nanometer SPIO tracers shown here in electron microscopy. We had to build our own MPI scanner. Now you can buy them from 2 companies, but we had to build them when we started on this 10 years ago. And the excitement is really illustrated right here with this GI bleed model versus a control animal. And so you can actually appreciate this red hue here is showing the extravasation of particles injected into the tail vein, and you can actually see them extravasating from this gastrointestinal bleed. You can see in the control animal absolutely no uptake. And so we did a digital subtraction angiography from the first scan to get rid of the circulating particles, and we were able to see this gut bleed with beautiful contrast to noise ratio. The X-ray is just an anatomic reference. So the highlights of MPI are it's got 0 radiation, very quick preparation time, which is actually really important, and no half-life, both of which are really important competing with nuclear medicine. The signal-to-noise ratio is amazing. The micromolar sensitivity in minutes, which is not as good as nuclear medicine, but in terms of dose-limited sensitivity, it's already competitive. The contrast and noise ratio is almost perfect because we have 0 clutter from background tissue and 0 attenuation within tissue. And it's quite robust. So unlike MRI, where you have a lot of trouble imaging in the lungs as well as ultrasound because of magnetic field tolerances, MPI only needs about 5% magnetic field tolerance. So it works anywhere in the body. And like I said, we're really competing more with scintigraphy and SPECT, not MRI. How does this work? So we have to do a little bit of a study of how Langevin physics works, which governs the saturation of these magnetic nanoparticles. So any field applied that's greater than about 5 millitesla gives you almost 100% saturation inside this nanoparticle to a remarkable 0.6 Tesla. And so this is an electronic magnetization. It's actually 22 million times stronger than what we image in a conventional MRI at 7 Tesla. Their paramagnetism effect is much, much weaker, of course, than the electronic magnetization in a magnetic nanoparticle. And so that is why we're getting this remarkable 1 micromolar sensitivity. And it's important to realize that unlike, say, hyperpolarized C13 or other methods that have hyperpolarization and then you inject, those tend to, those of course decay away to 0 very quickly over minutes or hours. This magnetization remains forever. So to make an image, what we do is we take a magnetic field gradient, much stronger than an MRI, and we actually slide it across the animal. If we had an SPIO in the left side of the brain and the right side of the brain, what happens is at t equals 0, you can see that the magnetic field gradient is negative for both of those particles. But as we slide it across, the magnetic field is going to switch from negative to positive, and then the SPIOs are going to adiabatically flip with that magnetic field traversal. And so the left side flips first, and then the right side. And so we see this in a voltage in a receiver coil, much like MRI, but not tuned. And then we can actually make an image by simply mapping time to spatial position because we know how that gradient is traversing the body. So this is the basis for so-called X-base MPI that we developed more than a decade ago in these papers here, where we formally showed that the point spread function of MPI is actually just this derivative of that Langevin curve. And so Currently, the nanoparticles we have about a 10 millitesla full with half max. And so in our very strong magnetic field gradients, we're struggling to get on the order of millimeter resolution. But I'm going to talk very soon about a breakthrough in MPI from my lab at the University of Florida, Carlos Rinaldi, where we're actually getting experimentally 1 millitesla full with half max. So we can actually get down to closer to 100 micron resolution. So MPI actually is illustrated here in this early image. And what I want to highlight is that in this helical phantom, we were actually able to do an equivalent to magnetic computed tomography to get 3D resolution. And then we displayed this as a MIP. We did a projection downward, and you can actually see this projection reconstruction data. And then we redid this with the entire phantom surrounded by tissue. And we didn't have to subtract. We didn't have to do any algorithms because that tissue is invisible. Totally invisible, and you get an almost identical image. So there's zero background signal in MPI. You get a linear positive contrast, totally quantitative at any depth. And like I said, infinite half-life to the tracers inside the body. The only half-lives you have to worry about are physiologic, not magnetic. So it's really ideal for both vascular and molecular and cellular imaging. And that's what I'm going to talk about today, these molecular imaging applications. Stem cell imaging, cancer imaging, and white blood cell imaging, which is really important for immunotherapy. Before I do that, I want to point out some in vitro studies we did a few years ago. There's a really unique feature to magnetic particle imaging. You saw that these particles had to rotate 180 degrees so that we could actually see them. There's two mechanisms, both Neal and Brownian, for that flip of 180 degrees. And if it's Brownian, we actually are very sensitive to the mechanical hindrance to the rotation. We can actually measure that time. And so without any detail, that allows us to actually measure the viscosity of the medium, which is kind of interesting. And it could be a very useful molecular imaging tool. And we've actually been able to easily measure binding. We haven't yet done this in vivo, but it is actually really exciting. One of the things we've been trying to do is see if we can actually use this to sense cell viability for, say, stem cells in vivo. And so more on that to come. So the first study on magnetic particle imaging and molecular imaging, we published a few years ago with Dave Schaefer, a prominent stem cell biologist at UC Berkeley. And you can actually see, we did a tail vein injection of pre-labeled cells, about 1 million, and you can actually see them trap in the 6 micron capillaries of the lungs. Not too shocking on day 1. By day 12, they were actually, they were moved by the RES system to the liver and spleen. And so the X-ray is just an anatomic reference scan. It's actually a CT study shown here, and this gives you the idea of it's a 3D study. So our sensitivity is about 2 nanograms, which translates to about 200 cells. But with these new particles, we think we can get down to maybe even single cell or 10 cells very soon. Here's a more conventional display of those studies, and you can actually see, again, the stem cells in the lungs on day 1 hour. And then day 12, you can see them migrate all the way to the spleen and liver. And if we just inject SPIOs without the stem cells, those are small enough that they go directly to the liver immediately. So you can read about this in Theranostics. Huge challenge is whether or not MPI can actually detect early stage cancer. Everyone here is aware that if we can detect cancer early, the 5-year survival rate goes from 25% for later stage detection all the way up to 98% if you can get it very, very early. And in fact, if you look at the deadliest cancers like lung and pancreatic, they're often diagnosed at later stages. So how could we actually target cancer? This is a big challenge, and many people have worked on this, not just my lab. And of course, nanoparticle targeting to cancer is a vast field. So what we did is we went with untargeted, long circulating nanoparticles. We weren't trying to have some antibody that binds to tumors. Instead, what we did is we just went with a 4-hour half-life tracer from Lodespin Labs, and you can actually see extravasation into this implanted tumor. It was 4 weeks post implantation. But you also see the challenge. Most of the particles are in the liver and spleen. But in this particular case, we actually got pretty good contrast for the tumor. But we would like something that's a little more robust than just the enhanced permeability and retention effect that was there. And so what we're hoping to do is to reproduce the imaging method from nuclear medicine called white blood cell indium-111, which is often used to image infection and maybe inflammation and maybe even cancer. So this is illustrated here. You can see a CT of this diabetic patient's foot, and then you can actually appreciate that there is an infection in this bone, and that's by using the patient's own white blood cells labeled with indium-111, which has a 2.8-day half-life. And so the fact that MPI has more of an infinite half-life could be a big advantage. So we just, in the early days of this, we published a couple of efforts trying to track, basically duplicate, indium-111 white blood cell imaging. We think this could be real exciting with cancer immunotherapy. We're working with Larry Fong at UCSF, a real leading immunotherapy guy. And so one of the latest efforts we just got accepted at Nanotheranostics is to use an antibody, anti-LY6G labeled nanoparticles. So we don't have to do the external labeling of the white blood cells, which is very cumbersome. Instead, we just inject it into the tail vein, and then neutrophils and macrophages take up those nanoparticles on their own. Then they go on to sites of myeloid cells in the bone marrow. So we're excited about this. The last 2 years, my lab has really been focusing on improving the spatial resolution of MPI, and we're getting very close now in vitro to 100 micron resolution. And this is not using a deconvolution algorithm. This is actually using novel super-paramagnetic iron oxide tracers instead of the traditional super-paramagnetic tracers. And we're getting not only resolution boosts, but also a significant SNR boost. All right, so we're super excited about this, and we wanted to figure out what the heck is going on. And so I've developed this chaining hypothesis that the particles are actually forming a chain. And we see that because that doesn't work unless the concentration is kind of high. And so that makes sense physically because the applied field is on the order of 6 millitesla at the flipping point, whereas the dipole field from the adjacent nanoparticles is 2 orders of magnitude stronger. And so just to show you that this magnetic field model of chaining actually matches pretty well. Here's our simulation of the dipole chain plus the Langevin curve, and we see a very good MH curve correspondence between real-world data and our simulation results. So we think we're on the right track. And in fact, our latest particles from Berkeley actually showed that we're getting about a 1 millitesla spatial resolution or magnetic resolution versus a typical 16 millitesla resolution when we actually are exciting this as a super-paramagnetic particle. And here we have a super-paramagnetic particle. And there we're just taking into account the true coercivity of our particle. So we're very, very excited about this. And just to put it in context, if we can reduce the cost of that gradient field by 10 times, that would actually reduce the hardware cost by a factor of 100. I've had a lot of experience building MRI scanners and MPI scanners, and so a 100-fold cost reduction is absolutely enabling. It also greatly mitigates the electromagnetic safety. The FDA, of course, has safety limits on both DBDT and SAR. And so finding a new nanoparticle, I think, is absolutely crucial to get a 1 millimeter human MPI scanner. And so I did some simulations on the magnet cost, and it actually would cost comparable to that of a 0.3 Tesla human MRI. And what I did here is real simple. If we had a 0.33 Tesla uniform B field that you could use in MRI, I made this whole body, and I just flipped the sign of one of the coils that forms this Helmholtz magnetic field, and you get about a 0.7 Tesla per meter field gradient. So that's, slightly compelling argument that we could actually reduce the cost of this to a low field MRI. So in conclusion, MPI has really superb sensitivity, already 1 micromolar, superb contrast and sensitivity. It already is competitive in many ways with scintigraphy and SPECT. And we are working now to radically improve the resolution, the only remaining physics challenge or engineering challenge with MRI. And we're seeing a 1D resolution and 2D of almost tenfold with an SNR boost as well. And we're really working to make this work in vivo. And ultimately, what we want to do is make this work in human MPI for a very affordable cost. Nobody has yet made a human MPI scanner. So just want to acknowledge all the funding from the NIH and others and answer any questions.